Okay, I'm going to ask you to quiet down because I can feel that I'm going to lose my ability to project here. Okay. So on Monday, we began reviewing the idea of mitosis. Who can quickly remind me what mitosis is? Yeah. Good. So it's reproduction of eukaryotic cells. Specifically, it is the asexual reproduction of eukaryotic cells. What's that mean, asexual? When we say that it's asexual reproduction, we mean what? Yeah, we end up with a clone. We end up with daughter cells that look identical to the original parent cells. In prokaryotic cells, we just call this process binary fission. It happens very simply. The cell can simply elongate, replicate its DNA, and then simply split in two. Why do we need a slightly more complicated mechanism in eukaryotic cells? Yeah, they have a more complicated sort of mechanism, and so this is a slightly more complicated process, and so we call the eukaryotic process mitosis. What are the 4 stages of mitosis? Yeah. Prophase, anaphase, metaphase, telophase. Good. You've been drawing this out every night, I hope? You should. Because it's knowledge we need in here. Today we're going to work with this, and we're going to talk about a variation on mitosis. It's going to be a lot easier to work with if mitosis feels comfortable and natural and like something you've just talked about in your own head. But let's talk through it quickly. In prophase, first of all, before we even enter mitosis, what has to have happened? What happens at some point during the regular life cycle of a cell, which we call interphase? Towards the end, before we can replicate the cell, before we can make a new cell, we need to make sure we have duplicated the DNA. And that doesn't happen during mitosis, that happens before mitosis. Makes sense. When we are exposing all the DNA, unzipping it and making a copy of it, we want that to happen safely in the protection of the nucleus. We don't want that to happen, in the exposed cell. So once we enter prophase, what's the first thing that happens? Yeah.
The nucleus dissolves.
Good. We get the dissolution of the nuclear membrane, the losing of the nucleus. Once we lose the nucleus, the DNA is going to need to protect itself and set itself up for easy movement across the cell. What's going to happen to the DNA? Very good. It compacts very, very tightly into these compacted chromosomes, right? Coiled, supercoiled again, to the point where they are visual under a light microscope. Tightly packed. No longer easily accessible for reading and writing and building, but that's okay because right now the focus is replicating the cell. Why do they look like an X? It's the 2 copies and they're stuck to each other. What is the central structure by which they are held together? The centromere. Good. Very good. All right, what happens in the next stage? What is the next stage? Yeah? Metaphase. All right, so after we've lost the... Well, that's okay, we'll get here. What happens in metaphase?
The 2 centrioles form and they grab the chromosome.
Yeah, and in fact, the centrioles form in prophase. I should have mentioned that. That's the I forgot. But by the time we get to metaphase, the centrioles have produced what? Spindle fibers, sometimes also called astral rays, but I don't like that term, but they are. And they actually connect to the chromosomes. By now, where have the chromosomes made their way? To the center of the cell. Tomorrow we'll get on the microscope and we'll actually look at some examples of this in real cell cultures. And you'll see that you can identify metaphase because you can see the chromosomes basically lining up perfectly down the center of the cell and the spindle fibers attached to them. Why are the spindle fibers from the 2 poles of the cell attaching to the chromosomes? It's going to pull them apart. What's the name of the stage of mitosis when they actually do get pulled apart? Prophase, then metaphase, then anaphase. Good. In anaphase, the spindle fibers shorten, and so the centrioles pull on the centromeres of the chromosomes, which holds the 2 copies together, splitting them, and now you get 1 copy going that way, 1 copy going that way. How many chromosomes does a human have? When we're done, how many chromosomes over here, how many chromosomes over there? 46. Is that 46 unique, independent, individual chromosomes? No, because we have 2 copies of each. What's the word for that? Diploid. Good, with diploid. So you have 23 chromosomes. Chromosome 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 21, 22, and then, well, we'll get there. And then you have one copy of that from mom, biological mother, and one copy from biological father, right? For a total of 46. All right. And then, once they are separated to the two sides, what happens in the next phase? And what is the next phase called? Telophase. Good. Sometimes called telophase. I'm not sure which is right. I think I say both. We get the nucleus forming again. Two nuclei forming. One on each pole of the cell around those there. And then, and you start to get the splitting at the cleavage row, right? Cleavage meaning cutting into two. Very good. Does that feel all right? Be comfortable with that. If you're not comfortable with that, you got to write it out. You got to draw it. If even that doesn't help because you're confused by it, you might need to come get some help and talk through it. But we have our little diagram here. And of course, that page ends with a little diagram of it too. But to be honest, I kind of find this hand-drawn one in some ways easier to follow. And certainly is something you can recreate yourself. All right. Today, we're going to carry on with the logical conclusion of this. And surely this is the question you've all been asking. This will be our last handwritten note for this unit. You're welcome if you like doing handwritten notes. And you're welcome if you don't, because this will be the last one. If you need some paper, there's some in my office, just on the back. Feel free to use it.
Nice one. I know she didn't do this. All by yourself. We're the greatest dude all for you. Sorry. Bro. It's like, it's a big guy.
Am I going? Yes, I am.
Do you know what the veins are called that connect from your eye to your
So they're not veins, they're nerves. And yes, the optic nerve. Optic nerve.
I've had one of those that are like too narrow.
Interesting. Now, there are veins and arteries that connect to the eye too, but they don't go to your brain, like they just work their way back to your heart. But that's interesting. So that's what they think the issue is. I want to hear all about this. I need to teach this right now, but you got to tell me about it. Oh, wow. That's cool that they're moving quickly, at least. That's nice. Because sometimes these things feel like they can take forever.
I waited for the MRI for like 5 months.
Yeah, I've been waiting. I scheduled an MRI in April, and I am getting mine in December. So I know that feeling. It's like, geez, I sure hope my brain doesn't like pop off in the meantime.
Yeah.
But anyway, that's a different discussion about healthcare spending in Ontario, which is an important conversation, but not one for right now. Okay, listen up. Shh. Today we're going to talk about a process related to mitosis. It's also a good reminder that in biology we love to have names for things that sound very similar to other names for things. So we learned about mitosis in grade 10. We reviewed mitosis on Monday. Today we're learning about meiosis. Sometimes I see people pronounce it meiosis. I don't know. That doesn't make any sense to me. But whatever, so I'm going to... if you want to know how I pronounce it, there you go. Okay. Why do we need a second procedure? Doesn't mitosis do the job for us? Mitosis creates two daughter cells, so you start out as just one cell, and then mitosis turns that into 2, then 4, then 8, and you just keep going, and you end up with, this. What's the problem? Why do we need a second procedure with an eerily similar name?
Because some things might sexually reproduce.
Very good. In fact, not only do some things sexually reproduce, all plants, animals, and fungi, for example, sexually reproduce. So we're just going to review here organisms that reproduce sexually. are diploid. And please remind me what that term diploid means. What does it mean to be diploid? It means that we have two copies of each chromosome that are not identical. And again, remind me, where do those two copies come from? That's right, your biological mother and your biological father. Fun fact, the word identical starts with an I. Oh boy, I'm having a moment now. Identical. Has that ever happened to you? You just lose the spelling of a word as you're going? Maybe that's just me. For example, remind me with humans. Humans have two copies of 23 unique chromosomes for a total of? Very good. We're going to use a term here that we used the other day, but I'm going to try to reuse terms frequently so they stick in our brains. What do we call regular non-sex cells again? S word. Good thinking. Somatic. Somatic cells are all the cells in your body that are not sex cells. So your nerves, your brain cells, your muscle cells, your skin cells, just every regular cell. Each somatic cell, oh thank you Mark Book, has 46 chromosomes. Who was it the other day was pointing out that it would be really nice to have a short form for chromosomes? I agree. So for example, if we have a copy of chromosome number 1, we could have chromosome number 1 paternal, that's from biological dad, and we could have chromosome number 1 maternal, and that's from biological mom. And then we could have ... all the way up to like chromosome 18 maternal. I did paternal in red. And chromosome 18 maternal, and so on and so forth. And again, remind me, how many do we have? Good. 23 pairs. Yeah. So these, the pairs of the same chromosome, hey, I bet we have a word for that. And it's probably not one you know. These are called homologous chromosomes. Homo meaning same, and GOS refers to genes, so the ones with the same genes on them. So homologous chromosomes means that there are two chromosomes that contain all the same genes, but different versions of them. So we can say that homologous chromosomes have the same genes but different alleles. And we're getting very close to being at the end of new words for today. Chromosome number 1, P for, what am I putting P there for? Paternal, which just means from biological dad. And C1M, M stands for maternal, and maternal means coming from biological mom. Good. Okay, now I've introduced yet more words. Oh my God. Has anyone heard these words before? You've probably heard one, but maybe not the other. Yeah.
I've heard the other one because I read a lot about like cat genetic stuff.
Ah.
I've heard them.
Well, that's good. That's interesting, though. So genes we're pretty comfortable with. What is a gene? Yeah. It's a section of DNA, and specifically, it's a section of DNA that does what? Section of DNA that does what? Well, that codes for something. Does that make sense? It's a single section of DNA. It'd be the equivalent of if you look through a big book of blueprints, a gene would be like the one page that tells you how to build like this wall. And then another gene would be the page on that tells you how to build the doorway. Does that make sense? A gene is a section of DNA that codes for one specific thing, one specific trait. So let's define these terms. That's a highlighter. A gene is a portion of DNA that codes for a specific trait. And to just use a really simple, in fact, an oversimplified example, let's say we had a gene that coded for hair color. That means in every one of our cells, we would have some DNA, one of our chromosomes, and at a certain spot on that chromosome would be a big run of DNA that codes to make the pigment in your hair that codes for your hair color. But the thing of it is, so that's good. We all have pigment that we make that's in our hair. Nobody's hair here is perfectly protein colored. There's all some kind of pigment there. And yet, notably, just looking around this room, and I know some of you dye your hair, but some of you I'm sure don't, and yet I look around the room and I see that not everyone has the same hair color. What's that mean? Well, it means that this gene has different versions. Does that make sense? You can have a version of the hair color gene. By the way, there's actually like 3 or 4 genes that cover hair color, but we're going to use a simplified world. Does that make sense? Where you could have a version of this gene that codes for blonde hair, and a different version that codes for brown hair, and a different version that codes for black hair and red hair and all these things and all the kind of shades in between. Does that make sense? Different versions of the same gene. A gene that says, hey, let's make some pigment for this hair, and the different alleles say, well, if you have this copy, your hair will be brown. If you have this copy of it, your hair will be red. Does that kind of make sense? You could imagine that if you were making different blueprints for a car, when it comes time to like color the interior, you would have a slightly different set of blueprints if they were painting it with one trim package versus another, though it'd still be the same basic blueprints. It would still be all the same stuff on all the same pages. I know that's kind of a weak analogy, but does that make some degree of sense? So we'll define that. Alleles are specific versions of a gene. So if we had a gene that coded for hair color, we could have different alleles. You could have one that codes for black hair, another that codes for brown hair, another that codes for blonde hair, and another for red hair. Again, this is a slightly oversimplified explanation of that topic, but I think it helps kind of clarify what we're driving at. And you can imagine even there, you can get a lot of variation by the fact that you might get one allele from your mother, but you might actually get a different allele from your father. And now, the hair you have is going to be somehow a combination of those two things. We'll talk about that early next week. Yeah?
I was going to say, aren't there also like recessive genes?
Yeah, we'll get into all that. We'll talk a lot about how your genes translate into your actual physical characteristics, but not today. So, the last thing we'll say before we get into this is that in the process of meiosis, I don't want it, that sentence sounds awful. How about during meiosis, calling an audible here. Sorry. One diploid cell produces new sex cells, and unlike somatic cells, which are diploid, sex cells have only one copy of each gene, and we have a word for that. They're haploid. Haploid and diploid. I think monoploid would be a better word. Hap to me sounds like half, so double and half. I don't know, I find that confusing. But regardless, haploid means only one copy of each chromosome. So in a human, our sex cells have how many chromosomes in them? Just 23, that's right. Very good. So that means when procreation actually occurs, a sperm cell with 23 chromosomes ends up with an egg cell that has 23 chromosomes. And at the point of fertilization, the sperm cell enters the egg cell, you end up with a nucleus containing now 46 chromosomes, 23 that were in the ovum, which comes from mom, and 23 that were in the sperm cell, which comes from dad, and you end up with a full set of chromosomes, full 46, 2 copies. Fair enough? All right. So meiosis is the process by which that happens. I'm going to teach meiosis 2 days in a row. Today I'm going to teach it like bird's eye view, simply, because it's complicated enough that if you try to teach it all at once, I think it becomes a little overwhelming. Does that make sense? So today we're going to teach it just kind of from a broad perspective, and then tomorrow we'll get in and get a little more detailed. Is that fair? Try to stick with me, draw it through. Let's get to it. Is there some reason I'm dotting that I? Okay, the first thing I want you to know is that meiosis is basically just mitosis happening twice in a row, back to back. So it's basically mitosis happening twice, and there's just one tiny little change. When we get to it, we'll talk about it, and that one little change makes all the difference. I bet you could predict it right now if you thought about it, but we'll get to it. So this is why we said it's helpful to understand mitosis well, because when we get to meiosis, it's just a variation on it. So because it is mitosis happening twice, we cut it into 2 stages, meiosis 1 and meiosis 2. Cycle happens twice. So let's talk about that. Before we get to mitosis or meiosis, what do we call the regular life cycle of the cell again? Anyone remember? I word. It's okay if you don't. But if we have a nice, just happy looking cell here, it's got It's got a nucleus, and it's got a bunch of DNA in there, not yet coiled up. We're at the end of interphase is correct. Good. So at the end of interphase. And remind me, right at the end of interphase, what happens? The last thing we need before the cell can reproduce. Why did I make that blue? Didn't mean to make that blue. What has to happen to the cell before duplication? Yeah. That happens right at the beginning of mitosis. We'll get there. But before that, before we want the nucleus, which protects the DNA to dissolve, what do we want to do to the DNA? Very important. Yeah. Yeah, good. So at the end of interphase, the DNA duplicates. Doing good for time. And we have a diploid cell, which means we have 23 by 2 chromosomes. Are you comfortable with me saying that? What do I mean by that? 23 pairs of chromosomes. And keep in mind, because it's duplicated, each is stuck to a copy of itself. And I'm going to use the highly scientific phrase, stuck to a copy of itself. Okay, so take that, get that drawn. And then you just told me, Sophia, what's going to happen in the first stage, not of mitosis now, but of meiosis. We're going to start meiosis part 1. And the first stage of, I'm going to do that all caps. That's going to look way nicer. The first stage of mitosis and of meiosis is called prophase. So we call this prophase 1, which just means the first time prophase happens. And there is nothing different here. It's exactly like mitosis prophase. So what happens? 3 things, really. What happens to the DNA? The nucleus dissolves. I like that. What happens to the DNA itself? Yeah, very good. So the DNA packs tightly. What will they now look like? Yeah, they'll look like X's. Why do they look like X's? That's right, they're stuck to a copy of themselves. So we'll have all of our maternal chromosomes here. We'll have all of our, what did I put, 4? 1, 2, 3, 4. And they all look like X's. So the nucleus dissolves, the DNA packs tightly, and there's one more thing that happens. Very good, we get the formation of centrioles. And what do they begin to produce? There we go. Very good. So I'll say centrioles and spindle fibers form. Again, is that any difference than prophase and mitosis? It's the exact same. And if we look, we still have, every time we draw one of these, I'm going to keep track of chromosomes. We have 46, and they're duplicated. Right? That's why they look like an X. They're stuck to themselves. What comes after prophase 1? Very good. And this is also exactly like mitosis. There's no change yet. So far, there's no difference between mitosis and meiosis. So what's going to happen here? Prophase, nucleus dissolves, spindle fibers form out of the new centrioles, and we get the tight coiling of the DNA. What happens in metaphase? Yep. Good, so spindle fibers. I'm going to use exactly that language. I like that. I don't know why I put that big space there. Excuse me. What else happens? This is more a visual thing. It's not so much something that importantly happens, but we see it. It's very visible. Yep. Yeah, chromosomes line up at the equator. Again, is this any different than what we would see in mitosis? It's identical. And again, I'm just going to keep track. We still have how many chromosomes? Very good. And they are still duplicated. Okay, prophase, then metaphase. What would normally happen now in the process of mitosis? Anaphase. And what would normally happen in anaphase? Meaning what? You're right. The copies would pull apart, and you'd get all 46 of them moving here, and all 46 of them moving there, pulled apart, so now that they are unduplicated. 46 and 46 unduplicated. But this is where the difference happens. Instead, in meiosis, they don't split apart. Instead, it's like a little tug of war, and the whole chromosome either goes to this side or it goes to that side. So we're going to draw that now. After metaphase, what's the name of the next phase? And the first thing I'm going to say is we're going to remind ourselves that this is where it gets different. So and what we're going to say is that the chromosomes do not split. Rather, 23 go to each side, still duplicated. And we'll talk more about this tomorrow, but you end up with one copy of each chromosome going to each side. And as it happens, it's not like all of mums go to one side and all of dads go to the other. We'll talk, like I said, more about that, but you get a mix. So it's not like a biological male produces some sperm cells that are all of his dad's DNA, and then some sperm cells that are all of his mom's DNA. Does that make sense? Rather, every sperm is a mix of some of mom's DNA and some of dad's DNA. This splitting happens randomly. We'll talk about that tomorrow. We're not going to write it down for now, but I happen to have drawn it that way just so the drawing is at least accurate. The important idea here is that we now have a slightly different situation because we now have 23 duplicated over here and we have 23 duplicated over there. This is something we haven't seen before. In mitosis, we always have 46. It's just whether they're duplicated or not. But here, the way it happens, we end up with only one copy of each chromosome over here, only one copy of each chromosome over there, and they're still in that X configuration. They're still stuck to a copy of themselves. Does that make sense? And now, you're going to get telophase and cytokinesis 1, and what's going to happen there? Telophase and cytokinesis are not different than in mitosis.
Telophase, they just I don't squish. That's all I'm thinking.
That's okay. That's good. They do squish. They're going to split into two cells by kind of squeezing in the middle. Before they do that, what do they have to form a new one of? Very good. They're going to form a new nucleus and split into two cells. Okay, again, try not to be intimidated by my beautiful art, but there we go. And we'll keep track of the chromosomes here. We have 23 here, which are replicated, and we have 23 here, which are replicated. And are these perfect copies of each other? I mean, just looking at it, this has some of mom's chromosomes, but some of dad's. And this has some of mom's and some of dad's, and they're different. They're pulled apart. So we already have not made perfect copies of one another. These are not the same. They're not identical. All right. Now let me ask you this. Why isn't this a sex cell already? We've done it. We now have a cell that only has one copy of each chromosome. And this, if mom produced one of these and dad produced one of these and we put them together, now we would have a new cell that would have 46 chromosomes. Good. There's just one thing that makes this not quite ready to go into service as a sex cell yet. What do we notice about the chromosomes? We have 23. Good. But they are duplicated. We want cells that have duplicated DNA as our sex cells. I guess you could. There's no reason specifically you couldn't, but we don't. Sex cells don't have duplicated DNA. They start with just a single copy. So now we're going to go through the whole process again with both of these cells so that we can pull those duplicated copies apart. Does that make sense? So the second time we go through this, it behaves just like mitosis, and we're going to pull those two copies apart. Does that make sense? All right, we're going to write that. And this is immediately after meiosis 1. You two fighting? Don't fight. And I saw somebody in the note had used, and I like this, so I'm going to write it here. So they undergo the stages of mitosis again, and somebody had that written just as this. And I actually like that because, not a bad way to remember, prophase, metaphase, anaphase, telophase. So they undergo these 4 stages again. To split the copies of the DNA apart. And I'm going to do this without any writing. If nobody objects, we'll just draw it. Is that cool? Okay, so coming out of meiosis 1, we had this. And then who can tell me what prophase 2 is going to look like? What will happen in prophase 2? Well, what happens in prophase? Yep. Very good. So we get the dissolution of the nucleus. We get the formation of the centrioles and spindle fibers. And that DNA is already like nice and tightly packed. So there we are. And we're just going to say here that both of these have 23 chromosomes and they're still duplicated. 23 chromosomes and they're still duplicated. Are you fine with me just putting it like that? I should put duplicated. Otherwise you're going to look at this after and be like, what the hell does dup mean? And fair enough. Right? That's probably what I'd do. All right, what's going to happen after prophase 2? Metaphase 2. And what's going to happen in metaphase 2? Good. So the spindle fibers are going to attach right to the chromosomes. And where will the chromosomes be? In the middle, like all bunched up? Yeah, they kind of line up at the equator. And what's going to come after? Oh, sorry, I should keep track of my chromosomes. How many chromosomes do we still have, obviously? 23 and the duplicated. And here, 23 and the duplicated. And what's going to come after Metaphase 2? Anaphase 2. And I bet you can tell me what's going to happen in Anaphase 2. Anyone? What's going to happen in Anaphase 2? Yeah, this time it's going to do what it didn't do in Anaphase 1. It's actually going to pull those two copies apart. Oh. And we'll make a little note here and say, now the chromosomes do split. And then what will the last stage of meiosis be? What am I doing? I don't know why I started writing that. Yeah? Very good. Telophase 2 and cytokinesis 2. And I kind of pair those together because I can. So in telophase 2, we get the cleavage furrow forming, right? The pinching of these off into two cells. We should get the formation of a new nucleus, like that. Inside the nucleus should be and after that, we should end up with how many cells? 4 cells. And interestingly, those cells are genetically different from one another. And tomorrow we'll talk more about how genetically different they are from one another and how the cell makes sure that happens. But let's make sure we keep track of that here. Now we have 23 and 23. What did I forget? Yeah, so I'm doing that right now. Also, I've made these look like they're X's, which they're not, so I'm going to redraw that. Because that's not what I'm trying to draw there. These cells can then be sent off to be processed into sex cells. There's additional things that happen. There's additional ways your body prepares this. Obviously, in the case of the sperm cell, they go through the process of spermatogenesis, where they actually then develop tails and all the structures they need to sort of do their job. Ova undergo a series of preparations. So this isn't quite ready to be a sex cell that is used, but this is now a sex cell that is ready to be processed into one that can be used by plant or the animal or whatever. Does that make sense? Okay. Whew. So at the end we have what we call gametes, sex cells. Okay, now, tomorrow, I'm going to give you a nice diagram of this that's pre-drawn and all that, but I think there's real value in drawing this out to self. I really do, because I think otherwise this can just seem like a lot of words. But really, there's only one thing in this entire process that is ever different from mitosis. Let's take a look. I'll zoom back in. But in this entire massive note where we go through all the things that happen, there's only one single moment that is different from mitosis. What is that? In Anaphase 1 or Anaphase 2? In Anaphase 1, instead of the chromosomes splitting like they do in mitosis, they simply move to one side or the other, still duplicated. Other than that, everything is the same as mitosis. And then, meiosis 2 is actually exactly the same steps as mitosis. The only difference is now you're starting with a cell that only has 23 chromosomes. That's haploid. Does that feel all right? Listen, it's a lot of information to have thrown at you in 3 days. All right? A lot. Hands up if it feels like a lot. Feels like a lot to me. I don't know. Good on you guys, I guess. Yeah.
You just said you explained like a little note, like you just explained the only part and like the only phase that was different. Can you write that in brackets at
Yep.
the bottom?
I put it there and I'll put it at the bottom as well. Okay, that's actually a great point. That's not a bad idea to put a reminder of ourselves here. Reminder. In the entire process, Anaphase 1 is the only time where it is different from mitosis. Sorry, you may not know this. It may not look this way by how incoherent I sound on a daily basis, but the truth is I sit at home at night and rehearse how I'm going to say this stuff and then write it down for myself so I'm not flying off the cuff. Right? I rehearse. Sadly, this is what I look like practiced. You can only imagine what this would look like not practiced, but all of which is to say, if this sentence sounds tortured, that's because I hadn't pre-planned it. But there you go. Okay, listen. Shh. A lot of information. Oh, guys, sorry. When I said listen, I must have misspoken. I thought I said listen, but actually I said keep talking while I talk. Is that what I said? Yeah? Oh, sorry to give mixed messages like that. Anyway, it's a lot of information. Information dense, information rich, all right? Lots of crap to know. And this unit is like that. There's two ways that goes. If you just kind of keep up with it and force yourself to talk yourself through it, keep up with these reviews, pay attention while we talk through it, come for help if there's something you're not getting, draw it out tonight, draw it out again tomorrow, and this stuff starts to solidify. This unit's super easy because it just builds on the same stuff. It's like a pyramid that builds up. Okay, sit down. Sit in the chair. Sit in the chair and sit there. Go ahead. So as I was spielifying, it's a pyramid. This unit just builds up on itself so that the last thing we learn is related to the first thing. It's not something our last unit was kind of a little bit of this, a little bit of that, a little bit of all over the place. Genetics really builds up to a point. It just keeps building on the stuff we already know, which means you want to make sure you have a good, firm, secure foundation in this practice, which means you want to write it down. You want to practice it. You want to write out. And if you don't understand it, talk to each other. Talk to me. If I'm confusing, talk to Alastruck. If Alastruck and I are both confusing, talk to Trush. If Trush, Alastruck, and I are all confusing, go on Khan Academy. Like, do what you got to do. Make sure this stuff makes sense to you. All right? It's important. I don't claim to be a perfect explainer of things. I'm keenly aware that I'm not. But there's somebody out there who can make this make sense for you, I promise. It's not me. Find them. Make sure you get it. All right? End of spiel. Don't start standing up. I'll throw things at you. I'm looking at you. Sit. Sit down. Sit down. I did. Sit. Were you? Yeah, right over here. I have a stapler right here, and I actually think my tape got stolen last week, which I'm quite upset about. Yeah, I'm sorry I don't have any tape. I have sticky notes. Is that useful to you at all?
Yeah, I'll use it. Yeah, really cool. Thank you so much.
There you go. Yeah. Sure. Yeah, you bet. Oh,
I finished it last.
wonderful. Thank you very much. Oh, I should stop recording. That's what I should do. It's so humiliating that I record all these incidents of me just being grumpy and miserable at the front of the room. And yet, here we are.